Our next speaker is from Capricor. Capricor certainly represents a second generation, or some might argue a third generation, cell therapy for treating heart disease. What's interesting to me about Capricor is not only are we talking about an allogeneic product, but a product that was really born out of autologous work, but a business decision was made to go allogeneic. But also, when we think about the language of cardiology, that language may be incrementally improved, not from a mesenchymal cell, but from a cardiac cell itself. So to talk us through Capricor, because I understand that Linda is on vacation in some very exotic place, is Luis. Thank you, Luis.
Thank you. The difference that we can find between the different kind of cells that had been involved in the reparative pathways, especially with the cardiac field, can be founded in the origin of the cells. And the origin of the cells is going to be relevant probably because the pattern of factors that are secreted and different biomolecules, but also probably related with the tropins by the right tissue. So we know that the cardiac-derived cells has probably a higher tropins by the heart, and that's why maybe the retention is bigger there, and that's why they can produce more specific factors that are going to activate the reparative pathway in the heart. And probably these reparative pathways is not just activating the endogenous stem cells, because that's what motivates the transition from autologous therapy to allogeneic therapies. We know that the cells not stay longer after the administration in the patient, but they activate different kind of response that are going to be responsible of the improvement in the patient. And that kind of response are mediated controlling the fibrosis, so probably they are controlling the secretion of the different collagen by the fibroblast. Probably even more important than that is to control the inflammatory process that is produced after the infarct. And in that way, we know now that controlling the inflammatory response that is produced after an insult is playing an important role, not just in the heart, but after every insult that is produced in different organs. And that can be very relevant because it's not just, as we were talking before, that talk about the right moment of administering the cells. The right administration seems to be very important in order to obtain the whole regenerative response. If you inject, in our case at least too early, maybe you don't control all the pathway that you want to control. If you inject too late, maybe you are not able to produce also the complete effect of the cells. So that's the transition that we made.
Let me slow you down a little bit. And first of all, we're talking about allogeneic cardiocytes or myocytes.
We are talking about allogeneic cardiosphere-derived cells, so some kind of progenitor cells able to activate the reparative response.
And we're in this case, we're specifically talking about an ischemic episode or post-STEMI.
We are talking about post-ischemic event, but also we are talking about heart failure, advanced heart failure, ischemic or non-ischemic.
So the reason why I want to delineate between STEMI and heart failure is because the time window in heart failure is going to be very different than it is in STEMI.
Totally different.
So in the case of STEMI, what is the golden period? What's too soon and what's too late? You seem to be suggesting there's a sweet spot.
Actually, the results that we have with the dynamic clinical trial, in which we are talking about chronic patients, class 3 in New York Heart Association class, we have observed a clear improvement in ejection fraction. That's a big deal because actually that will reflect in an increase in the quality of life of these patients. And actually, we are able to reduce the class from 3, 2, or even 1 in more than 92% of the patients. So that's for the chronic heart failure. If we go to the post-ischemic, we are talking about 1 month after the infarct, until 90 days, around 3 months.
Wow. Okay, so I'm shocked. I'm shocked that you believe the window is 30 days after the infarct. Yes. Isn't it? Don't you think the homing signals, that the damaged area of the infarct, that on the peri-infarct zone, those cells are being overworked, and so help me understand what you think these cardiospheres are doing 30 days later. And they're delivered via the infarct-related artery?
Yes. Actually, we are talking, depending on the clinical trial, we are using the artery that was responsible for the infarct, or for instance, for dynamic or even for Duchenne cardiopathy. we are talking about 3 different vessels in which we can increase the dosage that we are injecting.
Okay.
The response is not going to be directly only on the cardiomyocytes, but also all the kind of response that is produced after the infarct. Once that we have the insult, the kind of response that is produced is quite complex, and there are a lot of cells that are involved. In a first way, probably there are involved macrophages, neutrophils, and T cells, and then they are able to recruit cardiac fibroblasts that will be responsible to make the secretion of the collagen in order to make the scar. So there is a really long response that is produced for a long time, even longer than 6 months, until you have established the final scar. So in every step of that process, you can interfere in order to prevent the final heart failure.
But if I'm allogeneic, off-the-shelf ready, and a patient's presenting in the ER, and let's say they're going to be, they're going to go up to the cath lab, maybe they're going to get a stent, blood flow will be restored. Why not deliver cells right then and there?
That's an interesting point. With our data, we didn't observe clear improvement if you just administer the cells right after in the infarct. And maybe it's because the kind of population of cells that you have there at that moment are not the kind of population that need to be modified by the cardiogenic stem cells. And probably that's why you need to inject the cells in the moment in which the population of cells that are going to be involved in that reparative response are already there, and probably are related also with the kind of inflammation that is produced after the infarct.
So, Luis, help me understand when I go back and I look at Capricor, and again, we're going to stay focused on cell therapy. What data does Capricor have that suggests proof of concept? If I remember properly, and forgive me if I don't, the caduceus data actually showed that you could increase cardiac mass. But
Yes.
help me tie the increase in cardiac mass to all of the other measures that show me that it's making a difference in terms of quality of life.
Yes, we reduce the scar size and we increase the mass of functional tissue. That means that we are obtaining clearly and reproducible regeneration. It's just not reducing the effect, but improving the amount of tissue that is going to be able to pump blood. That probably will repercute in having an increase in quality of life of patients, and it will increase the ejection fraction of the heart and producing an improvement.
A few years ago, maybe a year, maybe 2 years ago, J&J made a strategic investment into Capricor. Do you still have dialogue going on with J&J?
Absolutely. Yeah, we have a very close collaboration with J&J, and actually we are collaborating very close in order to scale up all the manufacturing process in order to be able to supply the market with allogeneic cells when at the right moment.
And just to help me recall, the investment in J&J, was that a strategic investment? Do they have right of refusal for partnering options?
They have right to exercise an options based on the data of the clinical trial that we believe probably for 2017 called All-Star.
So I don't know if you know this, but I was kind of shocked to be on the partnering panel, and one of our panelists is from J&J, and he actually left the venture division of J&J to go pioneer an islet transplantation company that's trying to treat type 1 diabetes. So clearly J&J has interest in cell therapy.
Yeah, they are very interested.
Okay.
And actually also I want to mention, because you told us something that is very interesting, we are probably the 3rd generation of the product, because now we are working with a cell-free product produced by the cardio stem cells, and that are called exosomes. And the advantage of the exosomes, as you mentioned, we move from the autologous to allogeneic cells, and that is because we think that most of the fat produced by the cells are regulated by the growth factors and the different biomolecules produced by that cells. So we identify that cardiosphere-derived cells are able to produce exosomes. There are vesicles that recapitulate most of the fats produced by the cells. And the advantage of this is that there are no living material. So the ability to grow to obtain that product, to store that product, even to lyophilize the product, will improve the ability of translate that to the market.
Right. So what I hear you saying is that the 3rd generation might be to take cells out of the equation and go right to the trophic factors delivered via exosomes. Is that?
No, that's not correct. I mean, we know that the exosomes are able to recapitulate most of the fats produced by the cells, but we see different application and different indications. Both products can be useful. And actually for the heart, we would think that still the cells have a lot to say. And actually, maybe because the administration route. The exosomes are really tiny, really small, the size of a virus, and maybe the retention in the heart is not so good as the cells, because the cells get trapped in the vessels. For the exosome, we are exploring different indications, and actually we have really good results in ocular indication, and actually with animal models, in acute keratitis model, we observe some reduction on the inflammation and improvement in the healing.
Okay, great. Thank you. So help me focus now on what events, what trials, what data will Capricor be working on that I should be watching over the next 12 to 18 months?
Yes, during the next month, we expect to finish the recruitment in 2 different clinical trials that are ongoing. That's All-Star and Hope for Duchenne myopathies.
Okay.
Yeah.
Mm-hmm.
And also for Q1, Q2 2017, we expect to have some data regarding the All-Star clinical trial. And at the end, also at the end of this year, we hope to have some.
And remind me, what is All-Star going at?
All-Star is a clinical trial based on the use of allogeneic cardiosphere-derived cells for the ischemic heart disease produced after a myocardial infarction.
Okay, good. And when that data should be in early 2017?
Yes, we think so, because we expect to complete the recruitment in the coming months before the end of the year, and then we will expect to have some 6-month results in the close.
And how many patients in that trial?
120.
Okay, 120 patients, and that trial will be fully enrolled, I would expect, pretty soon if you're expecting
Yes.
data.
Yeah, we expect to finish the recruitment soon.
Okay, good. And what's the endpoint on that trial?
The endpoint will be efficacy, although we're also evaluating, of course,
Efficacy
safety,
as measured by what, though?
based on ejection fraction, infarct,
Wall motion abnormality,
wall motions,
and systolic
abnormality,
volume.
systolic volumes, 6-minute walk, and NIHA test.
Okay, so this is kind of a proof of concept trial here.
Probably we can consider like a proof of concept. It's a
Okay.
phase 2 clinical trial.
Good. Remind me, what does Capricor have on their balance sheet today? About how much money as the last reported quarter? It's not your area.
It's not my area.
Okay, we'll have to look it up in the
I
SEC
have
filings.
The CFO is.
I
is
see
in the
Leland over there.
audience,
Leland, you want to
and
tell us how much money Capricor has? Okay. And so what other events or catalysts should I be focused on as an analyst around the company?
It will be important for this year regarding the exosomes platform, because we expect to be able to present preclinical data demonstrating the efficacy of these products in at least one indication, and we are planning to present IND before the end of the year.
Okay, terrific. Thank you so much for the rundown on Capricor.